Hi, this is Biology 1406 lecture over Chapter 3.2 through the end of Chapter 3. When we last met, we were talking about water and the different bonds that it participates in. Just as a reminder, oxygen and hydrogen are held together by a polar covalent bond because the electrons are shared unequally. Oxygen ends up with a partially negative charge, whereas hydrogen ends up with a partially positive charge. What's interesting about water is what happens when we get lots of water molecules interacting. So when lots of water molecules begin to interact, what we see is that one water molecule will interact with another water molecule through a hydrogen bond. And that's what we were discussing last time we met. To continue, we said that because of water and those charges, there are four properties of water that sort of become emergent when we get lots of water molecules working together. Those emergent properties that you should know about are that water is cohesive, it's able to moderate temperature, it expands upon freezing, and it's a very versatile solvent. And each of these properties occurs because water is a polar molecule. We talked about cohesion, that first property. Cohesion is when water molecules stick to water. This is really important for drawing water up a plant. We also talked about adhesion. Adhesion is when water sticks to other stuff that is also charged. Again, really important for all plants, all organisms that require water to go up. This is the way that we get the water up and the way that we keep the water from falling back down. Related to cohesion and adhesion is something called surface tension. This is a measurement of how hard it is to break the surface of a liquid. Surface tension is related to cohesion because it's these water molecules interacting with one another and the water molecules that are below it that create this invisible film. So this is our first emergent property of water, this ability for water to act cohesive. And this is really important to think about because, again, trees require this. Also, if we're something like a water strider, this would be how we survive in our environment. The second emergent property of water is that water is able to moderate temperature. As we think about this, we can say the following, this bullet point right here. What happens is water can absorb or release a large amount of heat with only a slight change in its own temperature. So obviously I mean something different here by the word heat than I do by the word temperature. So thinking about it again, what water can do is it can absorb a lot of heat, but its temperature will just change a little teeny amount. So let's talk through some of these definitions. First, what is kinetic energy? Kinetic energy is energy of motion. So if I'm here flapping my arms back and forth, that's a lot of kinetic energy. Heat is different than temperature because what heat does is it measures the total amount of kinetic energy that we have due to molecular motion. Thermal energy and transfer from one body to another is what we call heat. Temperature, though, is a little bit different. As we look at temperature, temperature measures the intensity of the heat due to the average, not the total, but the average kinetic energy of the molecules. So as those molecules start bouncing around more and as they're speeding up, then we're going to see an increase in temperature. So maybe an example would help. Let's imagine that I go swimming in the ocean this weekend. My temperature, if I'm nice and healthy, is 98.7. Okay, so that's my normal body temperature. But the temperature of the ocean might only be 70 degrees. So on average, my molecules are moving much faster than the ocean. So my temperature is higher than the ocean. If we think about me versus the ocean, though, who's bigger? The ocean. So because the ocean is bigger, there's a larger amount of kinetic energy, and so the ocean ends up having way more heat than I do. Related to this idea of water, how does heat move? Heat is going to move exactly the opposite way than what you probably assume. Heat is going to pass from a warmer to a cooler object until the two are the same temperature. So when you put ice in your glass, it's not making things colder. Instead, what's happening is those warmer molecules with more heat in them are going from the warm liquid into the colder ice cube. In order to measure temperature, we use our Celsius scale. You should have been exposed to this in lab. On the Celsius scale, you should be familiar with the following 4 benchmarks. 0 degrees Celsius is where water freezes. 100 degrees Celsius is where water boils. Also important, we've got 25 degrees Celsius. 25 degrees Celsius is an important value to know because this is what we often refer to as just standard room temperature. In lab, if you're asked to run an experiment at 25 degrees Celsius, you should go ahead and do this right on your lab bench. You don't need a special refrigerator. Also important is 37 degrees Celsius, which is important to us. 37 degrees Celsius is your body temperature. That's normal human body temperature. So be familiar with these 4 benchmarks in the Celsius scale. Related to this idea of heat and temperature is something known as the specific heat of a substance. The specific heat of a substance, as defined in this first bullet point, is the amount of heat that must be absorbed or lost for 1 gram of that substance to change its temperature by 1 degree Celsius. If we were a physics or a chemistry or a natural science class, we would actually make you quantify this in lab. However, here in biology, we just want you to be able to work with this number and understand what it's representing. So it turns out that if you calculate the specific heat of water, it turns out to be 1 calorie per gram degrees Celsius. And you're like, great, Professor Miller, what does that mean? Well, this is actually a really, really high value. The specific heat of most liquids are decimal points. For example, the specific heat of ethyl alcohol is 0.6 calories per gram degrees Celsius. What this means is that water is going to really resist any change in its temperature because of this high specific heat. You've probably seen this happen if the following has ever happened at your house. So you go to make a pot of spaghetti and you throw the metal pot on the stove and you fill it up with water and you leave to do something and you come back and you're like, oh, the pot isn't boiling yet. I must have filled it up with cold water or something. And you go to grab the pan where the water isn't boiling, but the metal is burning hot, right? The metal is going to be able to increase its temperature really quickly. As those molecules start moving faster in the metal of that pot, the temperature increases. However, the water is different. As the water molecules are increasing in heat, as those molecules begin to move around, that heat is being absorbed, is being absorbed, is being absorbed. And so the temperature is not really changing. So the water's not quite boiling yet. Water's high specific heat is related or is linked to the hydrogen bonds. So let's go back to our pot of boiling water. Heat gets absorbed when hydrogen bonds break. So if I've got this big bucket of water and I'm trying to heat it, all of that heat is going to get absorbed, break a bond, break a hydrogen bond, break a hydrogen bond, break a hydrogen bond. I'm going to have to break a bunch of hydrogen bonds. I'm going to absorb a bunch of heat before the temperature changes. This property is super important for everything that's alive here on the planet. The high specific heat of water really minimizes temperature fluctuations to within limits that permit life. So consider living in LA. You're right next to the ocean, you're right next to Disneyland. If you live in LA and it's the middle of the day, the sun is up high in the middle of summer and all of that sun is beating down, and what is happening is all of that heat from the sun is being absorbed by the ocean. Now the ocean doesn't start boiling because water can absorb lots and lots of heat and its temperature is only going to increase a little bit. And so what that does is it keeps it really pleasant there on the coastline, right? Like 75, that's a perfect temperature. At night, when the sun goes down, what happens? Actually, the exact opposite happens. So what happens at nighttime is now that the sun is gone, the water is able to release some of its heat, so some of that stored heat is released. The ocean doesn't freeze when it releases that heat because the heat is being released as those hydrogen bonds form. What happens at nighttime in LA? It's still about 70 degrees. It hasn't gotten very cold. And so that water is making those temperatures stabilize. This doesn't only happen on the coast, this happens across the entire planet. So during the daytime, we're not boiling And during nighttime, we're not freezing solid because water is able to moderate temperature. Related to this is evaporative cooling. Evaporation is when any substance transforms from a liquid to a gas. As the liquid evaporates, its remaining surface cools. This is called evaporative cooling. So you can think of it as the hottest molecules leave. So when you start sweating, right, you're sweating, those molecules on your face have a lot of heat, they're very hot. As the water evaporates, it takes those hot molecules with it, and what's behind is those cooler molecules. Evaporative cooling definitely helps stabilize temperatures. That's important for us. That's why we sweat. But we're not the only organisms that sweat. Many animals utilize evaporative cooling. Plants also do this. So again, important for biology. What's the problem if you live in Texas? Humidity. Humidity inhibits evaporative cooling because the air is already full of water molecules. So you've got these hot water molecules on your face with all of this energy, but they can't evaporate out because the atmosphere is already saturated with water. So now we've been through our first two emergent properties of water. Number one, water is cohesive. We talked about how that's really important for plants. And number two, water can moderate temperature to permit life here on Earth. The third thing that we want to talk about is that water expands upon freezing. Or another way to say this is that the ice floats. The reason that ice floats in liquid water is because the hydrogen bonds that form in ice are more organized, making ice less dense. So I'm down here looking at the bottom of the slide, and as we look at the bottom of the slide, you will see two pictures. We've got this one that's in white, and then this other one that's over here in blue. As we look at these two pictures, you'll notice that over here in the blue image, what we get is a lot of water molecules jam-packed into this circle. This represents what water looks like when it's in its liquid form. There's hydrogen bonds forming, but it is very dense. There's a lot of water in that circle. On the other hand, when ice forms, what we get is more space. So as we look at this white circle over here, we're getting more space. There's still hydrogen bonds forming, but it is more organized. There's more space between those hydrogen bonds, so this thing is lighter. It's less dense. Because of this, what happens when we look at water is that ice in its solid form will float on liquid water. Water is actually its densest at 4 degrees Celsius. And yes, this is cool that ice cubes float, but what does it matter for biology? It's actually really important in biology. If ice sank to the bottom of our rivers and our lakes and our oceans, all bodies of water would eventually freeze solid, and this would make life impossible on Earth. And I want to talk about this for a second. So maybe you don't really like to eat fish, right? So you're like, who cares if we kill all the fish in the ocean or in the lakes? Like, I'll just eat other stuff. It turns out that all bodies of water are full of a lot of microorganisms that are actually producing oxygen for us at really high rates. Plants definitely photosynthesize, they release oxygen, but it's really these aquatic microorganisms that are producing most of the oxygen. So if you freeze your lakes and your oceans solid, you remove all of these organisms, and very quickly we would run out of oxygen that we need to survive. So the fact that ice can float on the top surface of a lake or a river is really valuable for biology because it keeps oxygen in our atmosphere. Because ice floats, this is also important for many organisms that rely on ice in their various environments. Global warming is real, and it is having a profound effect on icy environments throughout the globe. What we see because of global warming is that glaciers and Arctic sea ice are disappearing, and this is a problem in biology because many animals depend on ice for their survival. It's not all bad news, it's also good news. It just depends on which type of animal you're looking at, but there are being changes to our ecosystems as the ice environment is changing. As we look at this picture up at the top, you can see in sort of the lighter font where we traditionally had Arctic sea ice up there in the North Pole. And then you can see where that sea ice was in 2014. So things like polar bears, walrus, guillemots, they require ice to either breed or to eat or to complete part of their life cycle. As we get less and less ice, they're going to suffer. On the other hand, organisms like phytoplankton, whales, some of our migratory fish, they're actually doing better because now that there's not this big ice mass blocking them, they can actually explore more areas. Great picture here down at the bottom that you can see of how much sea ice has declined probably within your lifetime. This is a major problem that biologists are dealing with and that ecosystems are going through. So we've now been through 3 of the 4 emergent properties of water. The first one is that water is cohesive. The second one is that water has the ability to moderate temperature. And the third one is that water expands upon being frozen. Next, we want to talk about step number 4. The 4th emergent property of water is that water is a versatile solvent. There are 3 important vocabulary words that you should know the definitions of: solvent, solute, and solution. 3 S words, we want to keep them separate, so let's start with solutes. When we talk about a solute, this is a substance that's being dissolved. This is like a salt crystal or a powder or the sugar molecule. The next molecule that we talk about is often the solvent. Solvents are the dissolving agents. So if you're making up a solution, maybe this is water or alcohol or chloroform. The final mixture is what we call the solution. The solution is a liquid that is a homogeneous mixture of substances. So for example, let's imagine I was making some Kool-Aid for dinner tonight. The solute, the cloudy, powdery stuff, would be the Kool-Aid packet and the sugar, right? Those are the chunks, that's the dry stuff. The solvent would be the water, and when I mixed it all together, the solution that I end up would be the actual Kool-Aid that I could drink. One particular type of solution that we often talk about are aqueous solutions. Aqueous solutions are those in which water is the solvent. Often in biology, we're going to be talking about aqueous solutions because water is a really good solvent. It's able to dissolve things. As we think about water, notice that I keep saying that it is a versatile solvent. I'm not saying that it's a universal solvent. So it's a really good solvent because it's able to form hydrogen bonds because of its polarity. Let's look over here at this picture that's on the side of the screen. So the yellow and green thing represents a piece of salt. So that's sodium chloride held together with ionic bonds. Water is able to dissolve ionic compounds very easily. Water has a charge on it. Ionic compounds have charges on them. Water is able to dissolve things with charges. Notice how the arrangement works though. When we look at the water molecules, it's all of the oxygen molecules that are surrounding this positive sodium cation. Or in other words, it's that negative oxygen, that negative oxygen, that negative oxygen, the negative oxygen, the negative oxygen. Those partial negative charges are surrounding the sodium ion and they're able to dissolve it. If we look at the chloride being dissolved, however, it's the exact opposite arrangement. It's all of those partially charged hydrogen portions of the water molecule that are surrounding the negative chloride ion to dissolve it. When ionic compounds get dissolved in water, like this picture that we have on the side of the screen, each ion gets surrounded by a sphere of water molecules. This structure that I'm circling with the red pen is what we call a hydration shell. So it's important to remember that water is going to be able to dissolve ionic compounds. Water has charges, it can dissolve things with charges. Water is able to dissolve other molecules as well. Water can also dissolve compounds made of non-ionic polar molecules. So remember, a polar molecule is one that has charges on it. So large polar molecules, things like proteins, can dissolve in water if they have ionic and polar regions. One example your textbook talks about is this enzyme called lysozyme. It's this big protein, and we're able to get it to dissolve in our tears because it has these ionic and polar regions on it that are able to interact with water molecules. Related to this idea, this fourth concept that we have, that water is a versatile solvent, is the difference that we should know between hydrophilic and hydrophobic substances. A hydrophilic substance is one that has an affinity for water. Affinity means that you like something. So hydrophilic, if we just break apart the word, hydro means water, and then philic means loving. So these are substances that love water. Hydrophilic substances are going to be charged. Because water has that partial negative and positive on it, it's going to love being near things that are also partially charged. So things that are ionic, things that are polar. Other molecules are described as being hydrophobic. Hydro means water. Phobia means a fear. So a hydrophobic substance is one that does not like water, that's afraid of water. These water-hating molecules do not have charges on them. This might be something like oil. Oil does not like to mix with water. Water has charges on it. Oil does not have charges, so oil is hydrophobic. As we think about how water is able to be a versatile solvent, we are going to start talking about solute concentrations in aqueous solutions. In biology, most of the chemistry is happening in water because most biology is happening in these aqueous solutions. You need to learn how to calculate the number of solute molecules in a volume of solution. This is called molarity. I guarantee that I am going to ask you molarity questions on quizzes and exams, and this is one of the concepts that I'm going to continually keep coming back to with our comprehensive quizzes and our comprehensive exams over the course of the semester. You need to know how we can look at a substance and know, oh, there's 10 pieces of stuff floating around in there, or there's 100 pieces of stuff. Like how many solutes are floating around in a given amount of volume? So let's just start by talking about what molarity is. We'll go little by little. Molarity, abbreviated with a capital letter M, is the number of moles of solute per liter of solution. So you can think of molarity as being the number of moles, sorry, it's hard for me to write here, that we have per liter of solution. So example, if you've got a 1 molar solution, it means you have a 1 mole of solute in 1 liter of solvent. I'd like you to go ahead and try to answer these two questions really quickly. See if you can do them before I tell you the answer. What would I mean if I said a 2 molar solution then? What do I mean by a half molar solution? Right? Hopefully this is pretty straightforward. A 2 molar solution means that you have 2 moles of stuff, of solute in 1 liter of solvent. What does a half molar solution mean? It means that you have half a mole of stuff in 1 liter. Okay. So hopefully that was pretty straightforward, no weird math there. You should just understand what molarity means. If I say you have a 5 molar solution, it means you have 5 moles of stuff floating around in 1 liter of solution. All right, let's go on. When I ask you molarity questions, this is the form they're going to take. We're going to spend the next couple of slides working through this problem so you can see how to answer them. These are very common interview questions. If you plan on getting a job in biology or in the lab, this is a very common interview type question. So here we go. How many grams of C12H22O11 are required to make 1 liter of a 2 molar solution of that molecule? In these questions, I will always provide you some information. You're not going to need to memorize this. I will tell you how much each of those atoms weighs. So the atomic mass of carbon is 12 daltons. That's how much 1 carbon weighs. The atomic mass of hydrogen is 1 dalton. And the atomic mass of oxygen is 16 daltons. So this is what the questions look like. I give you a molecule. I ask you to make a molar solution. And then I'll tell you the atomic masses of the individual atoms. All right. What do you do next? Like, where do you go? The first thing that we need to do when we're going to be given these questions is we're going to need to take the molecule that we were given and we're going to need to figure out how much does that entire molecule weigh. Not just the atom, but the whole thing. So here's what we would need to do. We are going to need to figure out the molecular mass of that molecule. The molecular mass is the sum of all masses of all atoms in a molecule. So for C12H22O11, we would do the following. There are 12 carbons. I told you in the problem the mass of each carbon was 12. So 12 times 12, that equals 144 Daltons. And now we're going to do the same thing for the hydrogens. So there's 22 hydrogens. The atomic mass of hydrogen is 1. So 22 times 1, the mass of all of those hydrogens is 22 Daltons. We're then going to look at the oxygens. There were 11 oxygen atoms. Each oxygen has a mass of 16. 11 times 16, then we get 176 Daltons. If you add up 144 plus 22 plus 176, it turns out that sugar's molecular mass is 342 Daltons. So what I've done right now is I've figured out how much this particular molecule weighs. If you have one piece of this sugar molecule, it's going to weigh 342 Daltons. This is impossible to weigh out. Even if you have the most precise scale in the world, you cannot go to the lab and pour out 342 Daltons. It's just impossible. It's too small. So, since I can't weigh out these really small things, these Daltons, instead, when I'm measuring out molecules, instead of saying, oh, I've got 1 molecule or 2 molecules or 10 molecules, we often do these in things called moles. Very important bottom point here. 1 mole is defined as 6.02 times 10 to the 23rd molecules. This is also known as Avogadro's number. Please know this number. You should know that 1 mole equals 6.02 times 10 to the 23rd molecules. Again, that doesn't help me though. Like, what am I going to do to answer this question that we started off with? Well, it turns out that Avogadro's number and the unit Dalton were defined in such a way that we can measure out substances in grams instead of Daltons. This is because of calculus, because of integration. We do not have to worry about the math behind this. We just get to benefit from it. So here's where we're at. We have figured out so far that 1 molecule of sugar equals 342 Daltons. We know that, but Daltons are too small. We can't weigh things out in Daltons. Because of the way that Avogadro's number and the unit Dalton were defined, we can simply do the following. Instead of trying to weigh out something that's 342 Daltons, we can just weigh it out in grams. It is very possible to go into a lab and measure out 342 grams of a substance. This is easy to weigh, and the following is true. If you weigh out the molecular mass of a substance in grams, that automatically equals 1 mole of sucrose. So in other words, if you weigh out 342 grams of sucrose, right, we took its molecular mass and just weighed it out in grams, you automatically know how many molecules of sucrose you have. You've got exactly 1 mole, or 6.02 times 10 to the 23rd molecules of sucrose. So let's go back to our original question. How many grams of this molecule are required to make 1 liter of a 2 molar solution of the sugar molecule? We figured out that the molecular mass was 342 Daltons. How do we finish off the problem? Instead of weighing this molecule out in Daltons, we're going to weigh it out in grams. If we weigh out 342 grams, this will automatically equal 1 mole. But let's look at what the question was originally asking. The question was asking for a 2 molar solution. Remember, 2 molar means 2 moles. So what are we going to do? We are going to take 684 grams of this particular substance, and we're going to dissolve this in 1 liter of liquid. This will then create a 2 molar solution. Again, I guarantee that I am going to ask these types of questions on quizzes and on tests and on exams as we work our way through the semester. So I really want you to feel comfortable with this process. I've got a practice question right now that I'd like to give you some time to work through. So I'm going to pause. I'd like you to just take a moment and try to answer this question before I go ahead and answer it. So pause the video right now, work it out, come back in, and we'll see if we did the same thing. All right, let's see if we can do this question. How would you make a 1 molar solution of sodium chloride? We're being asked to make a 1 molar solution. Remember, that means 1 mole dissolved in 1 liter volume. It tells me that the atomic mass of sodium is 23 daltons and the atomic mass of chlorine is 35.5 daltons. The first thing I need to do is I need to figure out the molecular mass of sodium chloride. How much does a piece of sodium chloride weigh? And so it's 23 for sodium and 35.5 for chloride. If we add these up together, our mass is 58.5 daltons. Again, we can't measure things in daltons, they're just too small. So what do? We do the wonderful thing where instead of trying to measure out 58.5 daltons of something, we instead measure out 58.5 grams of something. That 58.5 grams automatically equals 1 mole. So we're going to take that 58.5 grams of salt and we're going to dissolve it in 1 liter of water. And that will give us a 1 molar solution of sodium chloride. So this sort of takes us through our 4 emergent properties of water. Number 1, water is cohesive. Number 2, water has the ability to moderate heat and temperature. Number 3, water is going to expand upon freezing. And number 4, water is a very versatile solvent. The fact that water is a versatile solvent is very important because most biology happens in water, and this is how we're going to dissolve proteins. This is how we're going to dissolve enzymes. This is how we're going to dissolve hormones. This is how things are going to move throughout a body, throughout cells, throughout living things. The next concept that we want to talk about are acids and bases. So hydrogen atoms in hydrogen bonds between 2 water molecules can shift from one to another. Here's the one that I'm talking about. This guy right here. This yellow hydrogen atom. This hydrogen atom is in a hydrogen bond, and what happens is sometimes the hydrogen atom is actually going to leave. When it moves, what happens is the hydrogen leaves its electron behind and is transferred as a proton or a hydrogen ion. These are synonymous. When I say proton or hydrogen ion, or you see this little H plus sign, those are all talking about the same thing. The new molecule that we end up with is known as the hydronium ion, although it's often represented in H plus. Sorry, I just drew my arrow on the wrong piece. Ooh, sorry, this is the hydronium ion. The molecule that lost the proton, this second guy, is now called the hydroxide ion. This is not something to worry about. This is just something that water does. We're just describing what happens to water. This is pretty rare. Most water molecules are not dissociating but there is a small amount of water in your glass of water or in your bottle of water where this has happened. And again, this is just called water dissociates. Another way to talk about water dissociating is to use this equation. Sometimes we say pure water breaks apart or dissociates into hydrogens and hydroxides. Okay, so water just pops apart. That's all this slide is trying to say. The fact that water pops apart or dissociates has a really big impact on biology, on organisms. If you're changing the concentration of hydrogens or hydroxides, this can drastically affect the chemistry of the cell. So in pure water, the following is true. The concentration of your hydrogens is always going to be exactly equal to your concentration of hydroxide ions. Some reminders: when you see brackets around a molecule, this means concentration of. So as I'm reading this equation down here, we read it as the concentration of the hydrogen ions is equal to the concentration of the hydroxide ions. And this is what pure water looks like. Everything's equal. When we add solutes, chunks, to water, these are called acids or bases, they can modify this relationship. So instead of everything being equal, when you add an acid or a base, it causes these concentrations to change. We define an acid as anything that increases the hydrogen ion concentration of a solution. So if, for instance, you take something like, let me choose a color here, hydrochloric acid, and you dissolve it in water, hydrochloric acid is going to dissolve very well because water is a versatile solvent, and you're going to get hydrogen ions floating around, and you're going to get chloride ions floating around. What this does is it increases the number of hydrogens in relationship to the hydroxides. So acids are going to increase the hydrogen ion concentration. A base is any substance that is going to decrease the hydrogen ion concentration of a solution. For example, we've got this picture down here. Let's imagine something like sodium hydroxide. Sodium hydroxide is a strong base. Sodium hydroxide, when you dissolve it in water, it breaks apart into sodium ions and hydroxide ions. The hydroxide ions, because there's so many of them, what they do is they interact with the hydrogen and they form water molecules. What is this doing? As those hydroxides are interacting with the hydrogens to form water, this is decreasing the number of hydrogen ions. Please be really careful when you talk to me about what a base does. Do not tell me that a base adds OHs to things. That's not the way to think about it. What is our base doing? It's reducing the hydrogen ion concentration. Here's why it's really important to think about it in this fashion. Let's talk about what happens when we add ammonia, which is also a base to water. So here's ammonia. It's NH3. If you add ammonia to water, what happens is it likes to accept extra hydrogens. So if you add it to water, it'll pick up this hydrogen right here, and it'll turn, instead of being NH3, into being NH4+. We haven't added any OHs, but this thing is still considered a base because what's happened? We've reduced the number of hydrogen ions. So acids increase hydrogens, bases decrease hydrogens. And then a neutral solution, something where we've got pure water, the concentration of our hydrogens and hydroxides will be the same. The pH scale is a way for us to quantify these relationships. In any aqueous solution, oh, we know what that means. What does aqueous mean? Yeah, any solution where water is the solvent. In any aqueous solution at 25 degrees Celsius, okay, we know that, right? That means room temperature. So in a water solution at room temperature, the product of the hydrogens and the hydroxides is constant, and it can be written in this formula. The concentration of the hydrogens times the concentration of the hydroxides always equals 10 to the negative 14th. You should be able to use this equation. If I ask you to use this equation, I'm going to keep the math very simple, stuff that you can do in your mind. So here's what it might look like. For instance, if your concentration of hydrogens is 10 to the negative 6th, you can figure out what the concentration of the hydroxides is because you know that those two things together have to equal 10 to the negative 14th. If it's been a while since you've multiplied exponents, just as a reminder, you add exponents when they're multiplied together. So 10 to the negative 6th times something is going to give me 10 to the negative 4th. Or in this case, it would be 10 to the negative 8th because 6 plus 8 equals 14. Try this one. If the hydrogen ion concentration is 10 to the negative 3rd, what is the hydroxide ion concentration? Go ahead and decide your answer here. If the hydrogen ion concentration is 10 to the negative 3rd, what is the hydroxide ion concentration? We know these two things multiplied together have to be 10 to the negative 14th, so it turns out that the hydroxide concentration ends up being 10 to the negative 11th. 3 plus 11 equals 14. All right, that's the first relationship you should definitely know and feel comfortable with. Second one, the pH of a solution is defined as the inverse log of the hydrogen ion concentration. We'll talk about each of these values. First off, let's talk about the hydrogen ion concentration. pH is interested in looking at what the hydrogens are doing, right? An acid is increasing the hydrogens, a base is decreasing the hydrogens. It's a logarithmic value or an exponential value. So as you go from one value to the next, it's not just getting double or half, it's actually a 10-fold difference. So when you go from pH 2 to pH 3, that's 10 times difference. To go from pH 2 to pH 4 is actually 10 times 10 difference. That's 100 times different. And then there's an inverse relationship. We'll talk about the inverse relationship on the next page. I do want you to be able to work with this particular equation on a quiz or an exam. I'm going to keep the math easy. I will always do hydrogen ion concentrations on quizzes and exams and stuff in the same format that we were just looking at, 10 to some power. So something like this. Okay? You don't need to pull out a calculator. You don't need to panic. If it's been a while since you've done longs, let me remind you how this works. If you are trying to figure out something where you are taking the negative log of 10 to some power, right? I'm trying to figure out what's the pH of this thing where my hydrogen ion concentration was 10 to the negative 6th. Your answer is quite wonderful. Your answer is your exponent. What is the inverse log of 10 to the negative 6th? The answer is 6. Let's try another one. What is the pH of this substance where the hydrogen ion concentration is 10 to the negative 3rd? We just look at that hydrogen ion exponent. The pH of this thing is 3. So you should be able to use this equation with sort of these simple round numbers that I've shown. As we start looking at acids and bases then, acidic solutions are anything that have a pH greater than 7. So all of these things are acids. Basic solutions are those that have a pH greater than 7. So all of these things down here are bases. Most biological things are right near neutral, which is pH 7, but there are extremes. Because pH is based on a log scale, just remember as we go from one value to the next, there's actually a 10-fold increase. Note that there's an inverse relationship. When we look at something that's an acid, it has lots and lots of hydrogens, right? That's what an acid is. But the numbers are actually 2's, 3's. They're opposites. Lots of hydrogens, but little pH. That's an inverse relationship. Let's think about the same down here with bases. What is a base? A base is something that does not have very many hydrogen ions. When we look at the numbers, those bases are actually the 12s, the 13s, the 14s. So there's an inverse relationship. With acids and bases, you should know about buffers. Most living organisms really have a set pH that they like to maintain. Most cells like to remain close to pH 7. Buffers are substances, chemicals, that minimize changes in concentrations of hydrogens and hydroxides in a solution. Most buffers are a weak acid-base pair that can either accept hydrogen ions or donate hydrogen ions as needed. One important buffering system that you'll learn more about as you learn about it in A&P is this carbonic acid bicarbonate buffer system. What happens is carbon dioxide can combine with water to form carbonic acid. This carbonic acid can break apart to form bicarbonate and hydrogen ions. This is how living systems are able to maintain a pH. If it gets too acidic, it can accept some of those hydrogens. If it becomes too basic, it can then donate hydrogens to maintain a set pH. Note, buffers are not trying to neutralize substances. They're not trying to automatically keep everything at pH 7. For instance, you have buffers that work in your stomach that work great at keeping everything about pH 2. So buffers are just trying to maintain some set pH. As we think about this carbonic acid bicarbonate buffer system, we want to mention acidification. Things that you do that burn fossil fuels, so think flying in airplanes, driving a car, turning on the lights, things that where we're burning natural gas or coal threaten water quality. When you burn fossil fuels, this releases carbon dioxide. Carbon dioxide, as we saw on that previous slide, can combine with water to make an acid. What happens when we put a lot of extra CO2 in the atmosphere here on the planet, some of that CO2 is getting absorbed by the oceans. The oceans are serving as a big buffer system for our planet. So we make all this CO2, it's out there in the atmosphere, and some of that CO2 is going in the oceans and becoming an acid. This is problematic because your water is actually becoming more acidic. But secondly, it's a problem because that is going to shift that buffering system so that less carbonate will now be available for organisms such as corals that require carbonate to build their outer shells. And so what we get is coral bleaching, which is happening across the world, and there's nothing really we can do about it because these organisms are just in too acidic of an environment and there isn't the carbonate available. And so we get these really sad pictures. Here's a nice healthy coral reef here on the left-hand side, and letter B and letter C are coral reefs where ocean acidification is happening. You can see that we're really losing the corals and the vibrance and the liveliness in those situations. This gets us to the end of chapter 3. Hopefully it helps. If you have any questions about chapter 3, please contact me. Email works great. Work through your study guide. Hopefully your sub was useful as well, and I will see you soon. Thanks.